Welcome, everyone, and thank you to our pharma partners for presenting, right? And thank you also for putting a low-key presentation together that is not talking pathways and things like that. So we are talking about updates. We are talking about progress in pharmaceutical development. And as I told you this morning, right, and as we spoke about it yesterday, this is really the highlight, the personal highlight for me, right, to stand here in front of you and show this full pipeline of commercial programs, and also this very, very strong showing in programs that are in the clinical testing phase. You might also remember that I really like soccer. I mean, real soccer, right? So, and in the past, we talked about common elements between drug development and soccer. But maybe today we also need to go a little bit beyond soccer, because I think the first lesson from soccer we all already learned. So here's again a short excursion into the German soccer league. And Harry Kane, he's the forward for Bayern Munich. And you see here, right, he takes a lot of shots and goals from very different positions. And we need to do that, right, in order to actually make goals. So that's really the common thing between drug development in a complicated disease and soccer. Don't panic. It stays at this level. So clinical development falls into four broad categories of biology. So going from the left to the right, we have the DNA with the genes, the DMPK gene, for example. And then we have a process called transcription where we make the RNA. And if we have the repeat, the repeat is present in the RNA. It misfolds and generates these hairpin structures, these loops. They look like lollipops, and that they act like ginormous traps for important cellular protein. And then when these proteins are no longer available, everything in the cells go kaput. So drug discovery falls into essentially the same four categories. So the first category interferes on the DNA level. Then we have interference with the formation of the toxic structure. Then we have approaches that target compensatory mechanisms, so approaches where we bring back the molecules that are trapped in these by these hairpin traps. And then we have compensatory strategies that are all the way downstream. And obviously, interference mechanisms that are all the way to the left, right, are mechanistically more specific. They, in theory, target the main mechanism of the disease, and they have broader side effects. I always liken that to, okay, if you have fever, right, you can have a fever because you have an infection, or you can have fever because you have a gunshot wound, right? Treating the fever doesn't heal the wound from the gunshot wound. It alleviates the symptoms. So that's how I picture these mechanisms. When we look at where the activities occur, you see what Charles Thornton calls the first wave, right? The activities of Avidity, Dyne, and PepGen, targeting the RNA-mediated mechanisms. Then we have ARTHEx compensating trapped proteins, and then we have Lupin, Harmony, and AMO in trying to compensate for the further downstream effect. Well, the cool thing is we have now also the second and third wave of programs. So we have for the first time Modalis trying to treat at the DNA level. And then we have novel approaches. We heard from Sanofi and their gene therapy strategies. But then we even have third waves, right? For example, where we have Vertex with a new generation of more broadly acting compounds, and we have Arrakis with orally available strategies. For DM2, we are a little bit behind. We are still more to the right, where we have strategies and potential to deal with compensatory mechanisms and potential complementation strategies for these trapped proteins. So soccer analogy 2.0. So there's really, really good evidence, right, that the disease, and we are all biologists, so or clinicians, and there's really good evidence to assume or to postulate that the disease variability is somewhat founded or based on the underlying biology. From the clinical trials and the preclinical data, we know that the drug entities are effective, so they work and they do their job. We also know from clinical trials and from preclinical data that the drugs get to their target. We know that there's very strong evidence that drugs get to the muscle and to other target tissues. So when we hear the anecdotal evidence that some people respond to the treatments and some people don't, we also hear, well, there's further evidence and opportunity for refinement of these drug candidates. However, if we say that, then we have to be scientifically rigorous, and we also have to state that there's a possibility of variability, right, in these clinical trial cohorts, and that we have not yet managed, right, to precisely stratify inclusion and exclusion criteria for these new molecular entities. So the new soccer analogy is that we need to go from mixed teams to teams of all-stars. And I liken this to the following schematic. So currently, we probably play with a combination of soccer player and American football players. And they're all excellent athletes, but they don't really play that well and great together. So what we need to do, we need to separate the soccer players from the American football players, right? And let they play in their respective teams. And I think that's when we get really exciting games and the best soccer and the best football that can be played. So the agenda for today, we do this in a two-part approach. So today we hear from Avidity, from Dyne, from ARTHEx, and Juvena, and Lupin Neurosciences. And then tomorrow from PepGen, Vertex, Sanofi, Modalis, and Arrakis. So, housekeeping. Please let me know beforehand if you want to give up your time or if you need extra time, especially our friends from Lupin. So please stick to your 10 minutes, and there will be no question and answer period afterwards. But please, the conference is an ideal venue, right? You all know the respective speakers to connect with them after. And without further ado, I'll hand it over to Avidity.
Thank you very much, Andy, and thank you for setting up the soccer as a great analogy. As I stand here and look over, I see such an amazing community that has come together, right? So it's an incredible privilege to be here and to be speaking to all of you today. My name is Kujo Bediako, and I get the privilege of being here to represent Avidity. And when I speak for the entire Avidity team today. With that, we have a little bit of a disclaimer. I think the pieces that I'll share is more of an evolving study, right? And we hope to bring this to fruition in the near future. I wanted to take a minute here really to pause and share our vision and mission statement with you. Like Avidity, we are committed to improving the lives of patients that are impacted with these diseases, including DM, through the development of new drugs, right? And you will see that very clearly in everything that we do. And one of the things that truly inspires me and has inspired me being here the last 2 days is the patients, the caregivers, and then the community around the patients and caregivers, right? You are our inspiration at Avidity, and you motivate us in terms of everything that we do. We started this journey really by focusing on 3 main studies. So we've run the Marina study. We've run also the OLE Alpha Marina, which is the extension study, and also the Harbor study. And we couldn't have done this without the people in this room and outside of this room and all the investigators as well too. So this slide is really to just say a thank you very much for being part of the incredible journey that we are on to hopefully bring new treatments for patients living with DM1. And just wanted to say thank you. It's still a journey that we are on, and you hear a little bit more as I go through it. And I will be mindful of time so that Andy doesn't, So the slide here is just to give you a sense of what DM1 is about. I believe that many of you are very much aware of DM1, but just wanted to highlight 1 or 2 things here. So DM1 is really a disease that starts with the respiratory system, but then it doesn't end there. It goes to the cardiac, and then you end up with some sort of a muscle weakness as well too, right? And these are really important because when you look at the studies that we are focusing on, we're looking at targeting the multi-systemic component of the disease and not just solving for one. And with that, we started to really think about how do we design a study that addressed the unmet need within the DM space. And we worked with MDF Foundation and also the clinical research team using the NDM1 study on the natural history to really understand what the disease is about, what influences that, and how can we ensure that whatever we bring out, it's making an impact in patients' lives, right? So that became the basis of what we started with. And then from there, we went into the Marina, like the phase 1/2 Marina study, to really understand what is going to be the optimal dose to help treat patients with DM1. And we identified that we saw an incredible data that I'll have a chance to share a little bit more. And that also led into opening the first randomized control trial that is potentially going to bring the first treatment to patients globally. With that, I just wanted to take a little bit of time to go through the phase 1 Marina study. We looked at 3 main cohorts, looking at a 1 mg per kg, looking 2, and then the 4. And through that, we established the 4 mg per kg as the leading dose that could really make a difference in the patients that we are serving. I'm going through it a little bit more faster to keep up on time. But here, one of the things I want to emphasize here is that in the Marina study, we treated about 37 patients. And you can see that we've done so far to date about 340 infusions, which is incredible. If you look at how far we've come on this journey. And among all of these, we have seen a very favorable safety profile. We had 1 SAE that resulted in discontinuation, but so far we've seen a very favorable safety profile. And here is just to give you a sense of what is the baseline for the disease, right? And if you are not actively treating the disease, what happens to patients? And you could see that, on the left side, you could see that it's either patients decline over time or they stay the same. And the one piece I want to show you on this slide is that when you look at the gray bars in terms of patients that are not getting any treatment, you could see a decline or staying the same. And you look at the purple bar, that is giving you a sense of patients that have gone on the Marina and the Marina OLE. You could see a clear difference after 1 year, right? So that is the value that we at Avidity, we hope that we can continue to add to patients' lives in terms of helping patients and their families to experience the life that they want. Moving on to this slide, and Andy, you might have to help me in terms of how to get this video. Okay, just to click it. Before I do that, when you go back to this, right, the gray ones, sometimes it's a little bit more easier to just look at charts, from a clinical standpoint, and you see things going left and right. But what we wanted to show on this slide is to really give you a sense of how what I have been explaining about and how deldeserin, which is our drug, it's making an impact in patients' life. And keep in mind, this is only one patient. Not all patients will experience this, but this is a true testament in terms of the benefits that patients can get when they go through treatment.
Ready? And squeeze, 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 and open.
You can see the patients have a very great improvement in their myotonia in terms of the hand opening. They are able to open their hand just after 1 year on being deldeserin. And that's a clear impact that we are having on the multisystemic of the disease. The next slide shows where we are now as a company and where we are with also like deldeserin. There are 3 main points that I want to make here, right? We are working at a lightning speed because we know how important this is going to be for the community. And we know the difference that it's going to make in patients' lives. So we are working at a lightning speed. We've engaged with the FDA, EMA, and also like PMDA to ensure that they are aligned on the study design, which will hopefully enable us to get this product to everyone globally for patients living with DM1. A very high level on what this study is about is it is a placebo-controlled trial, and we have one arm that's given deldeserin, and then we have another arm also that's given the placebo, and we are very well on our way to complete enrollment by the middle of this year, which will enable us also to get into a filing strategy with the FDA sometime very soon. Okay, with that, I just wanted to really pause here again and say a big thank you to this community. We couldn't have done this without you. We couldn't have even started this journey without the people in this room and the confidence that you've placed in us, the courage that you have, and also the support that you continue to give us as we go on this journey. So really wanted to say a big thank you from all of us at Avidity for everything that you are doing to support this study moving forward. We'd love to continue to be in touch, and we've got our QR code there. So please, if you have any questions or anything that we can do to help you, we also have a booth outside that you can stop to do that. We, of course, you are very aware also of the great work that MDF is doing as well too. So please feel free to reach out to MDF, and they will be much more glad to also help you as well. So thank you.
Great. Thank you very much, and thank you for your excellent time management. Next, we'll hear from Dyne Therapeutics and Ash Dugar.
Great. Thank you. So first of all, I'd just like to say thank you to Tanya, Andy, and the entire MDF organization for having us here. It's always a privilege to be in front of you and in front of this community. Thank you. I don't know what soccer is, Andy, but I'm looking forward to learning about it. These are forward-looking statements. Please refer to our website if you have any questions. So Kuja said it beautifully. We're here to listen to you, learn from you, and serve you in collaboration with the community, with the physicians, the scientists. We're here to try and develop therapies as quickly as possible and get them into your hands as soon as we can. So probably through the course of the meeting, you've heard a lot about the disease, so I'm not going to go into the details of it. You know it better than I do. Maybe one point I'd make is that this is a splicing disorder. At the heart of the disease, of this condition, it is the splicing or missplicing or dysregulated splicing. We call it a spliceopathy that really leads to the multi-system effects that Kuja presented so nicely. in all of your systems, in your organ system. So whether it's your skeletal muscle, including your breathing muscles, your cardiac muscle, smooth muscle, your gut, as well as the brain. It's really the underlying pathobiology that drives all of the symptoms in DM1. And so our goal is to develop a therapy that actually addresses that underlying pathobiology, which is the missplicing that occurs throughout the body, including the brain. So we have been advancing a compound called Dyne 101 into the clinic. It's in a phase 1/2 study, first in human study. And the construct of this is important. I'll just point out a few things. We have a Fab drug conjugate. What I mean by that is we have a construct that targets something called the transferrin receptor 1 or TFR1. And this is really important because it's highly expressed across all of these tissues that I've just mentioned, all of these different kinds of muscle components in the brain. And so we are using the body's natural cell biology to be able to drive our antisense oligo or our payload to attack the genetic basis of the disease, the toxic DMPK that resides in the nucleus. So it's really important that we're able to actually use this receptor-mediated delivery to drive that antisense oligo to get into the nucleus of the cells where it needs to go to be able to degrade the toxic DMPK, correct the splicing so that normal proteins for normal function are allowed. And so in this, we really see an early effect in myotonia, an early signal. And what we've learned from you and your physicians is this is important, but it's not the most important part of the disease. It's, we understand that folks learn to live with it, but it's really the broad functional improvement that needs to be driven towards to be able to have a therapy that's really meaningful. And that broad functional improvement in terms of muscle strength, muscle function, as well as quality of life issues as well. So before I get into the trial design and the results, I do want to acknowledge how much input that you've given us for the direct design of our ACHIEVE trial, our phase 1/2 trial. We've talked to key opinion leaders, KOLs all over the globe, not just neurologists, but physical medicine and rehabilitation specialists, cardiologists, pulmonologists, gastroenterologists. We've really taken a broad swath of these physicians who know the disease to help inform our trial. And very importantly, the patient community, advocacy leaders that sit here, as well as patient advisory boards, caregiver advisory boards, and have given direct input into the trial design, the most burdensome parts of the trial, and how to just support and educate families on what we're doing. So thank you. So very briefly, I'm not going to go through the entire slide, but we've completed what we've called the MAD or multiple ascending dose portion of the study. And after the 6-month MAD portion, which includes a placebo arm, all patients have the opportunity to go on to active drug in the open label OLE or LTE long-term extension. And in the ACHIEVE study, our primary endpoint is safety and tolerability. And we're looking at a number of other endpoints as well, including DMPK knockdown. Again, that's important to understand that you've engaged the target you want to engage. The splicing defect itself can be measured, as well as strength, function, and quality of life. There's a lot on this slide, so I'll point out just a few things. We believe that we have a very favorable safety profile with Dyne 101. We've seen no drug-related serious adverse events. The adverse events we're seeing are typical, largely mild to moderate in intensity. And I will point out that we have seen liver enzyme elevations in a minority of patients. Now, this is confounded by the fact that part of the underlying pathology of the disease is elevated liver enzymes in patients. And we've seen, even at baseline, before drug is given, 2.5 times upper limit of normal. But there's no liver dysfunction, no liver toxicity that we're seeing. And I'm really pleased to say that we've had nearly 900 doses administered and over 72 patient years of follow-up. So this has been dosed in a number of participants, and we've really been happy with the safety profile. So in terms of some of the data that I want to go through, here you can see the target engagement with the DMPK knockdown on the left-hand part of the slide, and then the splicing correction on the right hand. And the color scheme will be the same in all of the next data slides. The gray bar is placebo, and the TL bar is going to be Dyne 101 at the 6.8 milligram per kilogram dose. And that's our go-forward dose in our registrational expansion cohort. So I'll be focusing on that particular dose and dose regimen. So what you see here is about a 30% knockdown of the DMPK in this cohort, which shows we're engaging the target as we should, and about a 25% splicing correction as well. And our preclinical data predicted that if we could get to 20 to 25% splicing correction, we should see functional improvement. And we're going to see just that. And really, it's that functional improvement that we really want to focus on. Here we have myotonia, and you can see in the teal bar, the 6.8 mg per kg, we have about a 40% improvement in the myotonia as measured by the video hand opening time. So we see this improvement at 3 months and deepening of effect at 6 months. So we see this functional improvement in myotonia at this dose. And we also wanted to look at other measures of muscle strength and function like QMT total for strength, 10-meter walk test, as well as 5 times sit to stand, which is a test you may not be familiar with, but it's really important to understand lower limb strength, dynamic balance, stability. And so what we see across all of these measures is that the 6.8 milligram per kilogram dose, we see an improvement out to 6 months in all of these measures. And again, seeing this functional improvement across a number of measures, this totality of evidence is giving us a lot of confidence as we move into our registrational expansion cohort at 6.8 mg per kg. So this slide is really helping us to understand how does splicing predict functional benefit. And just to maybe give you the high level message here, what we're able to show is that we can show a change in splicing at 3 months, and that predicts functional benefit at 6 months. So we feel really good that splicing is something that can be used as a surrogate biomarker for efficacy because it's predicting clinical benefit. And again, this is across a number of measures where we're showing functional improvement based on that splicing change. We also wanted to look at patient-reported outcomes, specifically the MD-High. It's a measure which has 17 subscales that looks at nearly every aspect of myotonic dystrophy type 1. And you can look at it as a total score, which you have in this slide, and there's 17 subscales that you can look at in a valid way. And we did that, and I'll show you some of that data on the next slide. But here you can see a tremendous improvement in the patient-reported outcome, over 40% improvement in the MD-High total score from baseline. And then we pulled out some specific MD-High subscales that are related to CNS manifestations of the disease, such as cognitive impairment, sleep, fatigue, etc. And so what we're seeing across the board with these CNS-related aspects using the MD-High subscales is an improvement from baseline on a number of measures. And we know that what you've taught us is that the fatigue, the sleepiness, the cognition have huge impact on your quality of life. And so it's really important for us to be able to tease this out. What's not here is some underlying preclinical data that shows that our conjugate gets into the brain of non-human primates or monkeys, as well as the mouse models that we use. And so just really in summary here, I'd like to say that, we're advancing Dyne 101 into our registrational expansion cohort. We've selected the 6.8 mg per kg dose, and we're really excited to continue the progress of our study and report back soon. Thank you.
Great. Next, we hear from Dr. Walker from ARTHEx Bio. The floor is yours.
Thank you, Andy, and hello, everyone. Like the preceding speakers, I'd like to thank you all for giving me the time to speak and tell you a little bit about what we're doing at ARTHEx. Thanks to the MDF for organizing this session and all the hard work that has gone into it. If you go, do I advance the slides? I do. Sorry.
The green button.
Yep, got it. Okay. So let me tell you a little bit about ARTHEx. For those of you who don't know us, we are a biotech company that is based in Valencia, Spain, although we have people around the globe and we function internationally. We are a clinical stage company, which means that we are currently dosing patients in a clinical trial, and we are moving our lead program, which is that of ATX01, that's the name of our drug, in myotonic dystrophy type 1. This is a study in adults with type 1 myotonic dystrophy, and I'll tell you a little bit later about what we're doing in type 2 and plan to do in children as well. So let me talk about our lead program. And again, like the others, I'd like to thank you for, that's better. I'd like to thank you all for your input. We've spoken to numerous patients, caregivers, patient advocacy groups, and along with other specialists and experts in the field. Before we go into our study per se, I want to give you a little bit of the background. Now, there's a lot of science behind what we're doing, and we pride ourselves on being a very scientifically oriented company. There's a lot to digest around the science, and if you want more information, we have a number of posters here. My colleague Diego is here as well, and we can answer any of your questions around the science behind our drug. One thing I do want to point out about the disease that others have also pointed out is that this is, of course, a multisystemic disease. And one of the things people always ask us is, well, it's all very nice and well that you're treating the muscle symptoms, but what are you doing for the heart? What are you doing for the GI tract? What are you doing for the brain? And it is important that we treat the whole disease and not just the muscle. So the other thing that we always hear about is that there are so many trials out there, so many potential drugs. As a patient, do I choose what study to go into? How do I decide as a caregiver what's best for my loved one? And that's a difficult thing. But I think it's wonderful as well that there are so many options available at this time. So let me try to tell you a little bit about what makes maybe our program a little bit different. And there's two things. The first is how our drug works, what we call the mechanism of action. And the second thing is how we deliver the drug to the tissues that are affected. And with all of my colleagues here today, they'll talk about what their drug does and how it's delivered. And that's where the differences come in. And I think it's great that there are these differences. So in terms of how our drug works, we talk about the target. What are we actually working on? And what our drug is working on is something called microRNA 23B. And microRNA 23B is abnormally increased in patients with myotonic dystrophy type 1. And what we do is we block that. And what happens when that's blocked is that it increases MBNL protein levels. And as you know, MBNL protein levels is one of the things that regulates splicing. So by blocking MIR23B, we increase MBNL protein levels and we improve splicing. The other thing that MIR23B does is when MBNL1 protein is increased, it actually leads to release of MBNL from the DMPK toxic RNA foci. So we talk about a dual mechanism of action. We release MBNL and we separately increase MBNL. How we deliver the drug to the brain is by linking it to oleic acid. And oleic acid is actively transported into brain, heart, diaphragm, muscle, and other tissues. So people ask us when we talk about our dual mechanism of action, and I apologize, this is maybe a bit complex, but they ask us, well, how much of your drug effect is due to increasing MBNL and how much of your drug effect is due to releasing and decreasing DMPK and releasing the MBNL? So we did a little study, and what we looked at is we said, okay, well, let's compare our drug to a drug that works purely on DMPK. And this is a drug that's in green called a gapmer, and it's a drug that's no longer in development for various reasons. But we looked at our drug in the blue and we compared it to the gapmer, and we gave a dose that led to roughly the same amount of reduction of DMPK. So we adjusted the dose, so we got about 50% reduction of DMPK. And what we found that when we gave both drugs at a dose that led to 50% reduction of DMPK, we had a much greater splicing rescue. And as Ash said, it's all about rescuing splicing. So this means that in addition to the reduction of DMPK, there's something else going on. And that something else is the overall increase in MBNL protein. Next slide. So I'm going to talk a little bit about some animal models. Now, many of you have heard about these different animal models, and I won't go into the details of them, and each one has their pros and cons. You will have heard about the HSALR model, and very simplistically, this is a model where mice have muscles that are affected, and with a picture that looks a bit like muscles in human DM1. The other model I'm going to talk to you about is the DMSXL model, which is a model that resembles more a brain dysfunction that is seen typically in children or persons who have early onset DM. So we looked at both. And one of the things we found is that the drugs seem to have beneficial effects in both, including effects on improving splicing and increasing MBNL. And what you can see on, what's the pointer here? What you can see here is that we showed increase in MBNL levels in different tissues, including skeletal muscles of your limbs, your heart, your diaphragm, and the brain. We also showed improvement in grip strength, and that increased with increasing doses, and we showed improvement in splicing as we expected. When we looked at the DMSXL model, again, this is a model with many more repeats and is more like congenital DM1. And what we found here too is that we showed increase in splicing rescue with the drug. We showed decrease of DMPK in different parts of the brain, and we showed improvement in how the mice behaved. So that's how our drug behaved. And what we've done is we've taken our drug, which is an oligonucleotide, and we've combined it, as I said, with oleic acid, which is what allows the drug to get into the tissues of interest. And that's how we came up with our molecule ATX01. And by doing this combination, we can increase the stability of the molecule, make it safer, and allow it to be distributed to the tissues we want. We did a whole host of animal studies also to make sure that it was safe before we went into humans. And I can tell you that there was nothing of concern there. And we have quite a large dose range that we can administer to humans based on our animal studies. The other interesting thing to note is that because we're blocking a microRNA, and because this microRNA is the same sequence, that, in other words, the same order of chemicals that is seen in humans, as seen in monkeys, as seen in pigs, as seen in rats and mice, we use the exact same molecule throughout our development. So this brings us to, with all of this data, with input from your persons like yourselves, we designed the phase 1 ARTEMIR trial. This is a first-in-human study, so the main goal is to establish the safety of this drug in human patients. We are also looking at whether we have increases in our biomarkers, such as MBNL, increase in the muscle and splicing improvement, and we're looking at some outcome measures of how well the drug works. For instance, things like the amount of myotonia, things like the time to walk 10 meters, etc. We have two parts to this study, a SAD and a MAD. SAD means single ascending dose, MAD is multiple ascending dose, and in each part we have several cohorts. In the single ascending dose, by definition, we give a single dose, and then we follow the participants for 3 months. We have 4 dose levels. So we have a study by cohort. In each cohort, each group of patients, we have 8 patients, 6 of whom are randomly given active ATX01 and 2 who are given placebo. We have just finished enrolling our first dose cohort. We are going to have a meeting of our data safety monitoring board in the coming weeks, and then we'll proceed to the next dose level. The MAD is the same concept, except we give 3 doses 2 weeks apart. One of the things people say, well, that's all well and nice for DM1, but what are you doing for DM2? And the interesting thing, I don't have time to go through the detail, but the interesting thing is that based on our mechanism of action, it's likely that our drug will work in DM2 as well, the same drug. So our next plan is to look at DM2, which those of you in the audience who have DM2 will realize it is a somewhat different disease, even though you still have issues with reduced trapped MBNL1. And we are very committed to neuromuscular diseases, and this is giving you an idea of what we're looking at doing. We're also going to go into congenital DM1. Again, we keep getting asked about what are you doing for brain symptoms. We do believe our drug crosses the blood-brain barrier. We have seen in animal that we can show effects in the brain, so we are going to do a study in congenital DM1 as well. And on that note, I'd like to thank you for your attention, and please feel free to stop by our posters or grab myself or Diego if you have any questions. Thank you.
Thank you very much. So next we hear from Juvena and Dr. Modalis Youssef.
Thank you so much. Thanks, everyone. I'd first like to just start by sincerely thanking Tanya, Andy, and the entire Myotonic Dystrophy Foundation for the opportunity to present an update on Juvena's muscle regenerating fusion protein being developed for the treatment of myotonic dystrophy type 1. I'd also like to say that it's really these meetings and events that are so inspiring and really solidify our resolve, our commitment, and our passion to accelerating the development of drugs that can regenerate and repair tissues to rejuvenate lives. My name is Hannati Youssef. I'm a neuro and stem cell biologist by training and the CEO and co-founder of Juvena Therapeutics, which we launched in 2018 in order to map the therapeutic potential of stem cell secreted proteins and translate them into tissue restorative biologics that rejuvenate lives. All statements made today on our progress are forward-thinking and should be considered as such. At Juvena Therapeutics, one of our underlying theses is really in the development of medicine for movement, medications that can really promote better function of multiple organ systems in our body that are involved in enabling us to walk, to exercise, to travel, to play, and to be who we are. Specifically at Juvena, we are mapping proteins secreted by stem cells to different organ systems in which they are exerting tissue restorative benefits for the treatment of chronic diseases. The way that we really consider diseases is in the context of protein signaling. Signaling proteins in our body during the natural process of aging, as well as during stress, and in the case of genetic mutations like myotonic dystrophy, are dysregulated, leading to the loss of tissue health, what's known as tissue homeostasis, and the onset of degeneration that ultimately leads to debilitating diseases. At Juvena, our goal is to identify factors, really proteins, that can target these various pathways in order to promote and repair tissue protein signaling to rejuvenate tissues, ultimately leading to better tissue health and function. And that leads to the development of the first drug in our growing pipeline of biologics. And this is JUV161, which in many ways we consider the insulin of muscle. It is a modified form of a natural protein that's very important during embryonic development for muscle generation, known as IGF-2. And we have made much progress to date, including having received FDA orphan drug designation and approval by Australia's HREC committee to advance into phase 1 safety studies starting this month. JUV161 is comprised of a modified form of IGF-2 that we fuse to a larger, also naturally occurring molecule that circulates in our blood, human serum albumin, in order to improve its half-life and drug-like properties. It acts by activating AKT signaling in order to enhance metabolism, promote new muscle fiber formation, prevent muscle atrophy, as well as having anti-inflammatory benefits as well. We have developed it as a drug that can be subcutaneously administered, just like insulin is administered for diabetes, ultimately with the goal of creating it in pre-filled syringes that people living with myotonic dystrophy can take at home. And while I won't go into all the mechanisms and hallmarks of the disease, I think folks here have heard and learned a lot about that and know it very personally. I'll share the way that Juvena really looks at the disease, and we look at it in the lens of downstream dysregulation of protein signaling pathways. We know that the disease is caused by a CTG repeat mutation that causes an RNA hairpin loop, but what's maybe less appreciated is that the downstream aberrant splicing leads to different expression of genes and ultimately pathways such as those involved in insulin sensitivity, in metabolism, and in muscle survival and health. One of those core pathways being a downregulation of AKT signaling. This is the pathway that JUV161 activates by binding 3 receptors in order to restore signaling to more youthful, healthy levels, and by doing so, promotes muscle regeneration. And while there are many amazing therapies right now targeting specifically the RNA deficiencies and aspects of the disease, what makes JUV161 really unique is that we're going after the underlying core myopathy of the disease, the myopathy that occurs from development through growth and adulthood that leads to overall muscle wasting and reduction of strength. And so I will show just a few snapshots of some data that we generated both in human cells and mice. In human cells, we see that when we add JUV161, it enhances muscle regeneration to levels of even healthy muscle precursor cells, as shown in the right. And which one is the Okay, as shown here by the formation of these fibers, as shown in green. In a DM1 mouse model that we adopted from the Cooper lab from the Baylor College of Medicine, we were able to see that treatment of JUV161, even just for 3 weeks through subcutaneous underneath the skin administration, led to improvement in muscle weight. It led to an increase in the size of fibers, and it also reduced glucose, blood glucose levels indicative of anti-diabetic properties. Additionally, it causes a change in the fiber types in our body to more metabolically active fibers that are usually downregulated in the case of the disease and even with aging. And what's also exciting is that even in the face of the mutation still being there, we're not targeting the actual mutation, we see over time that the animals will increase strength, as shown here by grip strength, even while the control mice are continuing to lose function over time. And we also see, literally by measuring force output, an improvement of force and an increase in relaxation output. And so Juvena is now at the point where we've completed all the toxicology studies in order to now be able to start testing in people. We will have an announcement very shortly, but in the just next couple weeks, we will be treating healthy normal volunteers first, healthy individuals, in order to be able to test a few aspects, including safety, as well as the right dose by which we predict the drug will be efficacious. And we're hoping to see a few biomarkers that are indicative of that as well. Importantly, we're working closely with the leading clinical experts in the field, including folks like John Day, Tina Duong, and others, to design a study which we hope to start next year, where we will be treating folks living with the congenital form of myotonic dystrophy in order to see, after a few months of treatment, if they start to see the types of benefits that we've seen in human cells, in addition in animal models to date. This is the timeline, the aggressive timeline that we're on to do 4 phase 1 and a phase 2A study in the next 18 months, followed then by a much larger study where we'll be, very, very actively enrolling folks that want to have the opportunity to get treated with our insulin of muscle. With that, I would like to really thank the people at Juvena and the incredible community that we've leveraged to really advance, to discover and advance JUV161. from the leaders on our team who are currently very, very busy translating JUV-161 into the clinic, through to the many advisors we have, and a special thanks to many folks in the DM1 community, including Doctors John Day, Nick Johnson, Tina Duong, Melissa Hill, Andy Berglund, Tom Cooper, and so many others who we've been fortunate to work closely with, as well as the Myotonic Dystrophy Foundation, to get to where we are at today, and hopefully see our drug to people to rejuvenate lives in just the next few years. Thank you so much for your time and attention. And I would also be happy to share the slides if anyone would like to email me. So thank you so much.
Our last speaker will be Dr. Ala Sulaya Weidenfeller from Lupin Neuroscience. Take it away.
Thank you. Well, first of all, by the way of introduction, I wanted to start with a small preamble that actually I'm a newcomer, and although having spent a little bit more than a couple of years studying the myotonia and the impact of myotonia on daily life of people, I've never come across such a really strong organization as an MDF. I do come from the space of the rare disease, where I worked on different conditions, not in the neuromuscular, but with rare diseases. And I think the foundation that you have is really unprecedented. The level of the collaboration, the efficiency, the level of knowledge, and the desire to share the skills and to learn is really unprecedented and absolutely set for the success. So my sincere congratulations, and thank you for letting us be a part of this alliance. So let me introduce a little bit Lupin Neurosciences. We came from the field where our credo is bringing the affordable medicine, and the medicine is really in the innovation of bringing the generic innovation in the new form of the formulation, branded generics, and the biosimilars. However, in the recent years, we have been focusing on bringing these solutions to the neuromuscular space for people who suffer with rare conditions. And we had the opportunity to bring mixilotine back, which is a really solution for people with non-dystrophic myotonia. Is the condition that falls under the neuromuscular and channelopathies, but the only symptom that these people have is the myotonia. So I'm going to talk a little bit more about myotonia, and I actually skipped how do I get back? Back, right? Back. Just either one. I actually just kept quickly the disclaimer, but in my case is an important one. Just to remind everyone that the presentation is only for educational purposes. It's not meant to promote the use of our product because it's only approved in Europe for treating the patients with non-dystrophic myotonia. Talking a little bit about the myotonia, and of course, everyone in this audience is very well aware. This video is working. It's described when the muscles do not relax normally after the contraction, and it's a common symptom across the different myotonic disorders, including the myopathies. So next to DM1 and DM2, this so-called group of the channelopathies, which have a different presentation and mostly different triggers also, which is the stress, cold, but also dietary restrictions that would trigger and aggravate the myotonia. And I've learned really a lot over the last couple of years about the myotonia, because what we've seen is that it's often quite underestimated and also is not paid attention really, and under-perceived as an important symptom, especially in the group of people who suffer from DM1 and DM2 who do have more than myotonia, unlike in the channelopathies. So myotonia is not really a diagnosis, but it's a symptom that can affect people with both DM and NDM. And these diseases, they are very different. However, it's the symptom that feels very similar with muscle cramps, but also pain and hypertrophy, so the overdeveloped muscles. And it can affect different areas of the body, which does make it very difficult also to manage and address when it comes down to the clinical programs and clinical trials that we set up with your help. So it can also have different degrees of severity. And when it comes to the clinical trials, you may be surprised, but we didn't learn how to quantify the myotonia and how to be able to say that quantity of myotonia has this level of impact that we can really effectively measure. And that is a little bit of a challenge when we end up with so many therapeutic options on the horizon for people with all these different conditions that have myotonia as one of the symptoms. So I told you at the beginning that we have been focusing on bringing the innovation and innovative solutions in a simple way, affordable way to people with myotonia. And what we've done is we've looked into the ways of reformulating mexiletine, which is, of course, not a big deal. However, the drug on itself and the level that is required to achieve the therapeutic effect was not very easy to bring in a form that would be something beyond a capsule that you already know. So please come stop by at our poster that we have. It's I didn't look up the number, but it has a green color, unlike the others, so you can recognize it at the end of the poster hallway. And I can tell you a little bit more about the new formulation and the qualities that it brings. So that new formulation goes into a full clinical program that I'm going to talk just in a minute. I just wanted to share very quickly the outcomes of our pediatric investigator plan, right? That we have been conducted, and a part of this study have been completed with some encouraging results that will inform future pediatric dosing in these patients. So these patients were both non-dystrophic and dystrophic myotonic dystrophy, and we've enrolled them in a staggered way, starting with the older patients, and after informed pathology, we could continue with the younger patients up to 6 years old. I will not stop long here. All pediatric patients who have completed this so-called study 4, PIP4, have been offered and continued in the extension. It was a very short study, but we will have 2 years safety and efficacy data from these studies. Just to sum up on this, I don't know what happened because it does help when you see it a little bit. Doesn't matter. Just to sum up, thank you. The outcomes of the study were quite encouraging, and we will present the data in a different format, and also we'll share with the community the publication. We will present some posters also at the upcoming European Academic Neurology meeting and others in the pediatric meetings as well. But just to say, of course, it's not surprisingly, many of you know also mexiletine and also in children has been considered by 80% of the doctors as a good and a very efficient treatment. So really meeting the objectives of the study. What I really wanted to talk to you about today is the Hercules and Atlas study. We have about 12 sites across Europe and UK. These are the double-blind, placebo-randomized clinical trials where we will look into the safety but also efficacy of this new prolonged release formulation of mexiletine, and we're looking for about 80 people with DM1, but also DM2 patients are enrolled. It will be a smaller group of patients. So the Atlas is just an extension group, and as you have noticed, we choose the study names as the Greek heroic, really to associate the success of the study or the goal of therapeutic achievement as something that represents the resilience and improvement of the muscle function for people with myotonia. So this is a study plan, really in a nutshell. We'll have the 26 weeks of treatment, and there is an interim analysis which is incorporated into the study, and this is mainly with the purpose where we've learned, again, that the endpoints that we use in the clinical trials are not fully validated and then not fully also agreed between the regulatory and the payers, so the authorities who look for outcomes that really demonstrate the added value to the patient. So the myotonia improvement and how it impacts the patient's quality of life is something that we're looking in this study. So we are in the middle of the enrollment, and we're looking for considering also increasing the study size based on the interim analysis. So that's where when the half of the patients get enrolled and you look into the data in a blinded manner to decide whether you continue with the setup patient numbers or you would like to increase the study size. So the major study objectives is really to look into the efficacy and safety. And we have some additional secondary endpoints where it's important also to understand how the drug behaves, so the pharmacokinetics as well, and of course, things like the cardiac safety, etc. We go classically with the hand crimp relaxation time, and we need to really quantify it. So this is where the handheld dynamometer is used, and we support this study with the key secondary endpoints, which are more targeted towards the quality of life, which is, again, we don't have time to discuss this, but it poses a lot of difficulties of what regulators in US or in Europe also would be accepting to approve such a, not only this therapy. I think we all face a similar challenge. I just would like to finish with a summary of the scientific evidence and driving the scientific partnership in myotonic disorder research. And again, thank you, the community, but also the HCOs, our partners on the healthcare professionals, our medical experts who collaborate with us and who have co-created this, row of evidence demonstrating the importance of learning more about myotonia, addressing myotonia, and providing the solutions for this symptom, which is quite often overlooked. Thank you.
Well, let's give a big round of applause to the presenters, and until tomorrow. Thank you very much again for joining us here. So we have industry updates part 2. I won't give my spiel again. You had enough of me yesterday. So let's begin with PepGen, and Dr. Jenny Schaskis is telling about the updates from them.
Hello, everyone. Thank you so much for having us here. We can go to the next slide. So I'm from PepGen. We're a small biotech company located in Boston. And really what we're trying to do is be committed to transforming the lives of people with rare neuromuscular conditions. So people like Jubal and his family who have DM1. And how we're doing that is using something that we call our enhanced delivery oligonucleotide therapy, or EDO. So I know there's a lot of abbreviations being thrown around this whole week, but just one for us, the EDO technology. It's composed of 2 parts. One is a peptide. If you remember our name, PepGen, we have our peptide, and that's how we are getting into the different cells of interest. The second part is the drug part, or the oligonucleotide, and I'll tell you on the next slide how we work. So we know in DM1, and you guys have been hearing many times throughout this weekend, that it's caused by this repeat of the CUG. Now that repeat forms clumps, and those clumps act like a sponge to suck up something that's called muscleblind. Now the role of muscleblind in the body is to act like a proofreader. So you can see the sentence is maybe a little bit small, but it says, the sky, random word, is blue. So what muscleblind is trying to do is take out that random word so that you can have a normal sentence that has proteins throughout the body that don't have any errors in it. So our approach is using our molecule called PGN-EDO-DM1. And instead of knocking down DMPK throughout, we are just targeting the DMPK that has that expanded repeat, those CUG repeats. So we act by binding to those repeats, allowing the muscleblind, that proofreader, to go out into the cell and to correctly take out that wrong word in the middle of the sentence. And we hope we can do that throughout the body, including in the skeletal muscle, heart, digestive system, and respiratory system. So we would not be here today without all of the learnings we have from all of you. I'll talk a little bit about our clinical trials, but we have learned so much and have really designed these from your perspective. So some things we've done is taken your thoughts in terms of who should be eligible for trials. Thinking about on a visit day, how can we make things easier for you? We know fatigue is definitely an aspect of the disease and there can be long visits, but how can we make that easier? We also learn from you which symptoms we should be measuring and trying to reduce burden as much as we can. So that leads us to our two studies. So at PepGen, we have the Freedom study and then our Freedom 2 study. So the Freedom study is a single dose study looking primarily at safety and which dose we think is the best to move forward. Then we have our Freedom 2 study, and that's multiple doses every 4 weeks. And there, of course, we're looking for safety, but also looking at, are we having those downstream impacts? So who might be eligible for these studies? So you can see on the slides here, it's adult DM1, so 18 to 60 years old or down to 16 for our second study. You must have confirmed genetic testing. We do take muscle biopsies, so we require a certain amount of strength in the leg to make sure that we can do that safely. And we also make sure that people have some presence of the myotonia so that we can look if there's any effect of the drug. As I mentioned, we are not currently looking at people that have congenital DM1, or if you're on mexilotine or other agents specific for myotonia, we ask that you wash out of them so that in the study we can really see, is the effect of the drug or was it due to something else? And of course, we have a couple other things too, but these are the main ones. So going back to our first study, the Freedom study, again, this is just a single dose, and we're looking at around 32 people. So this drug is given by an IV infusion in the arm, and we're looking at different doses. So in each dose, there's 8 people, randomized 3 to 1. So what that means is for every dose group of 8 people, 6 will receive drug and 2 will receive placebo, and you won't know which one you're at. And of course, we're really looking for the safety of the drug, but looking as well to see, are we getting into the muscle and are we having some of those effects? So we're excited a couple months ago to have released the data from our first 2 doses. So we looked first at 5 and then at 10 milligrams per kilogram. And what we're seeing so far is a favorable safety profile. The most common things have been things like headache or nausea. We're seeing that we're getting into the muscle. So we look at that muscle biopsy and we see that the drug is getting in. The other thing is, really importantly, we are showing that we are taking out that word that's not supposed to be in that sentence. We have shown a correction of splicing, both at that lower dose of around 12%, and then at that higher dose around 29%. Now, if you have a good memory for numbers, this is similar to what people are higher than what people are seeing with repeat doses. And this is just with one dose. So we're really excited to see what this is going to look like with repeat doses and at higher levels too. So that brings us to our next study, the Freedom 2 study. And then this is IV doses every 4 weeks and at around 24 people. And we're currently enrolling in Canada and the UK and hope to also be in the US soon. So this might be a little bit small, but it's also on our website. These are all the centers that are currently enrolling for our trials, either for Freedom or Freedom 2. And I cannot go without saying a massive thank you to all of the participants that have played a part so far, all of our colleagues in the field who have advised us on how we should be designing our trials, and all of you for giving us feedback. We don't have question and answer time today, but we really do want to hear from you. If we haven't already, please come talk to us, because we want to learn from you and learn from each other and do better in the future. So if you're interested in learning either about the Freedom or the Freedom 2 trial, you can go to the website with the QR link there, or to our table, or email us at clinicaltrials@pepgen.com. Thank you all very much.
Thank you very much. So next we will hear from Vertex and Dr. Ziv Parker.
I want to thank Tanya and MDF Foundation for organizing such a wonderful, informative conference and giving us an opportunity to share our program and details of our study design. I'm Ziv Parker. I'm a neurologist and neuromuscular physician. I'm clinical lead for DM1 program at Vertex, where we are developing an investigational peptide conjugated oligonucleotide, or PMO, named VX670 for patients with myotonic dystrophy 1. Before I go into the details of clinical trial, I want to walk you over the design of our VX670, how it is designed to work, and why we believe that it has a potential to address missplicing. So if you see here, the green part is the PMO. PMO is the active part of the molecule. It goes and targets the expanded CUG repeater, expanded part of the RNA that's causing some of the manifestations of the disease or missplicing there. It is linked to two peptides. One is cyclic peptide and one is linear peptide. These peptides are specifically engineered to overcome the barriers for delivery of PMOs. We know that naked PMO have difficult time getting into the muscle. There are three main barriers: getting into the muscle tissue, muscle cell, then escaping the endosomes, and then getting into the nucleus. And these peptides help this PMO get into the nucleus where the expanded CUG repeats are, where you want to target that. So here in this slide, I'm showing how it works. So if you see up, I'm pointing to right side of your slide. So if you see, this is a cell membrane. The solid line is a cell membrane, and the small particles are the PMO. And here, the cyclic peptide helps this PMO go into the cell. And when it goes into the cell, it makes these circular structures called endosomes, and that are seen here. If other peptides are not there, this endosome will go take these material for degradation. So they will go and they will be either thrown out of the cell or will be degraded in the lysosomes. But because of these structures, these two peptides, PMO escapes the endosome, and now then it goes into the nucleus, a nucleus where you want to go. And here the nucleus is shown in the dotted lines. So PMO now with these two peptides goes into the cell, escapes the endosome, and goes into the nucleus. Why I'm telling you all these in such detail? Because in other diseases, we know for Duchenne muscular dystrophy, where naked PMOs have a lot of challenges in getting into the cell in the nucleus. And this is the strategy that we are using in this compound to overcome those barriers. When it reaches the nucleus, and that's on your right side of your slide, there's expanded CUG repeats here where the splicing factors are twisted. This RNA comes in between those repeats that you can see here in green color. It comes between those, the expanded CUG repeats liberates splicing factors, liberates MBNL1 that can resume its normal function, which is the splicing in dead cells. Okay. So in short, the SPX670 is a peptide conjugated PMO that is binded to two conjugate peptides that helps to overcome the delivery barriers of PMO, gets and engages with expanded CUG repeats in nucleus and corrects missplicing. And that's why we believe it has a potential to correct missplicing. That is one of the underlying cause of the disease manifestations. We are studying this molecule in a phase 1 study. In this phase 1/2 study, in this study, we're investigating its safety, its tolerability, its pharmacokinetics. Pharmacokinetics means how it distributes in the body. We're also studying how it affects splicing in these patients. This study is named Galileo study. This study has 2 parts. Part A is SAD, or single ascending dose part. It has been completed, where single doses were studied and their safety was assessed. Now we have moved on to second part, which is multiple ascending dose part, where repeat doses of drug is infused in these patients, and that part is ongoing. Patients who are adults, 18 to 64 years of age, who have genetically confirmed diagnosis of DM1 would be eligible for this study. All patients who are part of this study in these 2 parts will be eligible for an open label extension study where they will get the active component for a few years. Once the patients who are enrolled in these studies will be randomized to 6-to-2 treatment allotment. So that means that in the group of 8, 6 of them will be getting the active compound and 2 of them will be getting the placebo. In conclusion, our enrollment in single ascending dose is complete. Part B is ongoing. And this trial and the data collected from here will inform the future development of this compound for the therapy of DM1. Lastly, I want to thank MDF for this organization. And more importantly, I want to thank you. I want to thank patients and families. And your input was critical in designing this study. And with your participation, this study is possible. I also want to thank clinicians whose partnership has made this study possible. Thank you all.
Great. So now pay close attention, because now the new kids on the block present. So first we'll hear from Sanofi, Dr. Didier Hui, and their new gene therapy-based approaches.
Thank you. Thank you. Thank you to Tanya and Andy and the MDF to inviting us here. So that's my only disclaimer, which I am an employee of Sanofi, so that would be an easy one. Maybe you don't know, but Sanofi has been in the field of rare disease for more than 40 years now, based first with a company called Genzyme, and then Genzyme was merged with Sanofi, but we kept developing some of these therapies for rare disease. We've been interested in DM1 a few years ago. What we have tried to do these past 2 days, and I was incredibly. We listened and we learned a lot already in 2 days, and that's going to keep being that way for, we hope, many years to come. Okay, so gene therapy is not wizardry. It's a real therapeutic approach that use DNA as a vector, and that use also AAV, which is probably the scary part, adeno-associated virus. We are not infusing virus to anyone. We are not even using pieces of virus to infuse to anyone. What we do is we use this ability of these very little viruses, AAVs, that are non-pathogenic for humans. What we are using is some of the sequence of the capsid of these viruses, and we mimic it to encapsulate pieces of DNA that we use as therapeutic agents. So it's been used in the past for, there are several strategies, gene addition, which is, for example, to replace a non-working copy of a gene. Can be gene silencing. That's what we have been using here for DM1. There's also gene editing. This is not what we do. We are not modifying your genome. This is yours, and we are not modifying it. We are adding a property to silence some of the mRNA that are bothering you. What's really interesting is that we are infusing DNA, which means that it's a single infusion and then you're done, because the DNA is going to stay in your cells and keep making RNA, which is the therapeutic part of the molecule. It's a one single infusion, and then the effect is supposed to last for months or even years. It's been used in other pathologies like SMA, Duchenne, hemophilia, and the effect is still active after years and years after the infusion. So we've seen that 50 times since yesterday morning. DNA, which is what's wrong with DM1, that makes these long mRNA that are very sticky, and then that hence the problems that happen in the cells with MBNL1 and so on and so on. So our approach is to use one of the mechanisms that the cells use to defend themselves, which means that the cells don't like double-strand RNA. They like double-strand DNA and single-strand RNA. So what we do is to put a piece of DNA that codes for this piece of RNA that is going to bind to the mRNA. And the cells is going to degrade it itself and then get rid of this long DMPK mRNA that is bothering everyone. So for every clinical development, we have to do our homework and go through very specific steps. So it starts with the Petri dish level, which is with cells. And then if that works, then we move to a mouse model, a DM1 mouse model. Then we check for safety, and the closest to the humans are these non-human primates. These little monkeys where we test and be sure that whatever we are infusing and injecting is safe. And it's only when all these three steps are done and validated, then we can move to clinical development. So it's a little technical, but the Petri dish step, we've shown that it's target engagement, which means that in the cells, it does what the therapy is supposed to do. Then the mouse model, in addition, that was also interesting, is that some of the myotonia, which is in the mouse model, disappeared, and then their cardiac function got better also. And then the non-human primate studies, we showed that there is a good therapeutic effect because the humans have a DMPK gene, so we can assess what happens to that gene. And also, this was fairly safe. That was the main thing that we wanted to show. So based on all these checklists, all these boxes have been checked, and then we are now moving to the clinical arena, SAR 446268, which is the acronym for the therapy that we're using. Once again, one-time disease-modifying therapy. So we are opening these clinical studies because the Brave for the AAV, which is in the middle, to remind. So for you to remind it. Safety, tolerability, efficacy, but once again, mostly about safety. This is all the first thing that we look at associated viral vector, mediated gene therapy, participants aged 10 to 50 years with non-congenital myotonic dystrophy type 1. So the clinical design is going to be in two parts, part A and part B. Part A is called the dose escalation phase, which means that we start with a low dose of vector and see in around three participants, how the safety evolves on what's their liver function, what's their cardiac functions, how does the muscle function work. And then if this is safe, then we move to a medium dose, which is dose 2, and then once again to the dose 3, if once again this is safe. Once this part A is done, then we select a dose that we are going to expand in the part B with around 20 participants age 18 to 50 first, and then we're going to open it to a younger population, 10 to 17. All the patients are going to be followed for 2 years, and then 3 years of additional follow-up. That is a requirement by the FDA to follow every patient in gene therapy for 5 years, and the FDA has blessed the clinical design, so we are ready to start. All participants will be followed for 5 years, and then we'll see what happens. Thank you.
Wow, everyone is on time today. Great. So next we hear from Dr. Keith Connolly from Modalis Therapeutics. And Modalis is really interesting. If you paid attention yesterday, they are targeting the mechanism that is most closely to the causal mechanism. So they're trying to interfere on the DNA level. So take it away.
All right. Thanks for that, Andy. And so I think that this, instead of being an industry update, it's going to be more of an introduction, since this is the first time that Modalis has presented at the MDF conference. But, it's really been a real honor and very inspiring to see this group of folks, from patients to physicians to researchers, all coming together for a common goal. And I really sincerely thank the organizers of MDF and the MDF community for welcoming us in to tell you a little bit about what we've been trying to do for treatments for DM1. Okay, so let me get started. So we're Modalis Therapeutics. These are scientists working away at our site in Waltham, Massachusetts. This is just a few miles outside of Boston. And so at Modalis, we have been developing a molecular tool that is made of 2 components. We have a Cas9 protein that acts like a GPS. So this guides this tool to certain regions or specific regions of the DNA. And once this tool is found the location in the DNA, the other component is a gene expression effector. This acts like a dial to turn gene expression up or down. We're hopeful that this tool can be used to treat genetic diseases like DM1. And since this is our first time, sorry about that. Since this is our first time here, I'd like to introduce some of the biology that we're looking to exploit to make this work. So I'm going to take us back to some of our earlier biology classes in the next few slides. Okay, so as you all know, our bodies are pretty complicated. They're made of many distinct organs. We're made of some 30 trillion cells with something like 400 different cell types that all have specific properties and perform various functions. But what's interesting about this is that each of these different cell types are all encoded, or all have the same instruction manual encoded by our DNA. So a question that biologists have been studying for a long time now is, how can one instruction manual direct the development of so many different cell types and organs in our bodies? Let me go forward. So the complete answer to this is still being worked on, but it seems like this has a lot to do with how our DNA is packaged in our cells, which is also pretty remarkable. In fact, as an analogy, and since we're in Indianapolis here, if you took the DNA from just one of our cells and it made it into a string that wrapped around the Indianapolis 500, or the Indianapolis Motor Speedway a few miles from here, you would need to be able to pack it into the size of roughly this bouncing ball here to mimic how our DNA is packaged in the cell. So our DNA lives in a really crowded space, but there's still a lot of activity, there's still a lot of activity that's needed to express genes directed by our DNA. This is done by making RNA and protein that give the cells the properties to help them perform their function and give them some of their properties. To do this, there are also many protein factors that help organize the DNA and organize gene expression as well. Let's see. Okay. Some of these proteins can even add chemical tags onto the genome, and these tags can affect the conformation of the DNA, also the activity of the portions of these DNA. This can impact whether these genes are expressed or turned on, or if they're silenced or turned off. This process of influencing genes, gene expression and turning it on and off has been termed epigenetics. So this means on top of genetics. So these are sort of modifications to the instruction manual that don't change the underlying DNA text. One famous example of epigenetics that you've likely seen before is the fur patterns in the calico cat. So these alternating colors are the result of patches of cells that have different epigenetic profiles. So the patterns of how genes are expressed or silenced can dictate the properties of our cells. This can help explain how we can have many different cell types with just one genetic code. This would be like sitting next to a couple of people at the murder mystery dinner last night that have the same vocabulary, use the same words, or have the same genome. One person at the table might use words to indicate that they have a sweet tooth by expressing or silencing certain words, while another might give themselves away as a killer, while you look for a chair at a neighboring table as quick as possible. So just as these different usages of the same words can make these two very different dinner partners, our cells can have very different, can be very different depending on gene usage. Let's see. So these are the genetic mechanisms that we're looking to harness and control to treat diseases by specifically altering gene expression by turning on beneficial genes or turning off toxic gene expression. And we might think that we might be able to affect gene expression by targeting genetic differences in patients with DM1. One example of this would be suppressing DMPK. DMPK can have extended regions in the RNA transcript that make it more likely to aggregate. And when this aggregates, it can sequester some of the proteins that I mentioned before that can be important for gene expression. Our goal is to reduce DMPK expression through epigenetic modifications and prevent it from causing alterations in gene expression. To validate some of these ideas, we package these tools into viral particles that can deliver them throughout the body. And we tested them in a mouse that mimics a small portion of the genome of DM1 patients. After treating the mouse, we harvested various muscle tissues and found significant reduction in DMPK expression in all the samples that we tested, and almost all of the samples that we tested. We found that this treatment had a particularly strong impact on DMPK expression in the heart, which is an exciting finding for us. So we're looking to apply these tools to this and other genetic diseases, as well as sharpening and refining the tools as we develop them into therapeutics. So thank you for your attention, and here's some of our team members working and playing here. And one more thank you to the organizers and the committee for having us here at this nice conference. So thank you.
Well, thank you very much for this, especially these efforts to not talk in scientific jargon and making it high level. So exciting news, right? Hopefully you'll be back at the next conference and show how these things are actually working. So the last new kid on the block is Arrakis. And it kind of follows the same trajectory we've seen for RMA, for SMA. So where we started with an oligo infused into the spinal cord, and then we had gene therapy approaches, and then we had oral approaches. And Dr. Mahoney will tell us about their oral delivery strategies to treat DM. take it away.
All right. Thank you, Andy, and thank you to MDF for inviting us to give this talk today. My name is Sarah, and I am the biology co-lead for this preclinical DM1 program at Arrakis Therapeutics. So just to give a little bit of an introduction to our company, we're a small company in Waltham, Massachusetts. That's outside of Boston. A lot of biotechs are there, as you may have heard. And we're a platform-based company, which means that we're not necessarily focused on a particular disease area. Rather, we have a technology that we try to apply to human diseases that are amenable to those, to that technology. And so what is our technology? It's targeting RNA with small molecules. So how is that different from what you may have heard before with small molecule drug discovery? Well, just on the top of the slide, we have a cartoon, which is the basic central dogma of molecular biology. Basically, each cell has DNA within a nucleus. That gets transcribed into messenger or mRNA, and that mRNA is used as a template to make proteins. So traditional small molecule drug discovery has focused on targeting those proteins and pockets within those proteins to be able to affect either the function of that protein on, other proteins, or to affect proteins binding to other species within the cell. Arrakis is taking a different approach to try to, drug the undruggable by targeting not the protein, but the RNA itself. And as I mentioned, the DNA gets transcribed into RNA. The RNA is not just a straight line of nucleotide after nucleotide. It actually folds on itself, as you can see on the bottom cartoon, and it makes these base pairs. So G's pair with C's and U's pair with A's, similar to how you would think of as a DNA double helix. But it's not exact matches all the way across, creating these bulges and stem loops and three-way junctions that we think we can target with our small molecules. So how are we applying this to DM1? So we think that small molecules are a good fit for DM1, and it's a good fit for our technology because of the pathogenic mRNA that causes DM1. On the left-hand side is a depiction of how we might think of the molecular mechanism for DM1. You have this mRNA of DMPK with these CUG repeats at they call the 3' end, or just like the right-hand side, the end of that mRNA transcript. And in DM1 patients, they have an expansion of these CUG repeats from 50 to thousands. So what this does is inside the nucleus, where this is the cell nucleus, which is the organelle where all this is happening, this creates these, what they call aggregates, containing both these CUG repeat mRNAs and also proteins such as muscle-blind protein. What that does is it captures these muscle-blind proteins. They call it sequestering, and it basically prevents them from doing their normal function. The normal function for muscle-blind protein is mRNA splicing, which is part of a maturation process. That mRNA is not going to get exported and used and translated into proper proteins without proper splicing. So the sequestration and capturing of muscle-blind protein by these CUG repeats basically prevents it from functioning, which leads to downstream effects on the cells, then the tissues and organs, and eventually what manifests as symptoms in patients. So why a small molecule for DM1? So first of all, I want to describe the difference between a small molecule and a large molecule, or also known as biologics. So small molecules, first of all, they're smaller than biologics. They generally, you think of small molecules, you think of things like Tylenol or Claritin, or things you take over the counter orally. They're shelf stable at room temperature. And like I said, you just take a little pill. This is in contrast to biologics, which often have to be kept cold. They're often an IV route of administration, they often lack that broad biodistribution that small molecules have. Small molecules can often go everywhere in the body and can affect multiple organ systems, which is really important for DM1 because, as we've talked about many times, it's not just a skeletal muscle disease. It affects the CNS, it affects liver, it affects the heart. So we want to be able to help treat all organ systems for patients. On the right-hand side, here's a depiction of of how we're thinking about how our small molecules work. And so you have your RNA with all the bulges. Those bulges are these mismatches between base pairs. And in the different colored dots are depicting the splicing factors that are being sequestered. And we think that by bringing in a small molecule, it will displace those splicing factors and allow the splicing factors to go do their normal functions. We have a few slides of data just to show what our small molecules can do. The first assay that we want to show is our bread and butter assay. We have tested hundreds, maybe 1,000 compounds in this assay. And basically what it is we take a piece of CUG RNA, we take a muscle-blind protein, we mix them together in a test tube, and then we apply our small molecule and ask ourselves, does the muscle-blind get displaced? Can that small molecule separate those two? And we see that we can get that kind of separation from our small molecules, and we see that we are trying to build in more potency, which means that it takes less and less of a molecule to have the same function. So every iteration that we do on designing our small molecules, we're hoping to build in more and more potency. We're getting a little bit more complex, where we have a cell system. So these are muscle cells from a patient where we've applied either a vehicle control on the top row or our small molecule on the bottom row. And we've asked what happens to these nuclear foci, which are the aggregates, like I mentioned, where the CUG repeats can capture those muscle-blind proteins and stop them from doing their function. So on the top row, we've stained muscle-blind in green, we've stained CUG repeats in red. And on the right-hand side, where you're looking at an overlay, you can see the muscle-blind protein and CUG repeats appearing in the same dots. So that's from a patient-derived cell. When we incubate with our compound on the bottom row, what I hope you can appreciate is that the muscle-blind staining or in the green, you don't see those dots as well. It gets a lot more diffuse within the nucleus. And also you see a disappearance of that yellow staining, which suggests that even though there still are aggregates of the CUG, the muscle-blind is no longer bound to it. So now it can go do its own function, which is splicing. So moving along to asking whether the splicing can be rescued, we took those same cells, we applied our compound, we isolated the RNA and asked within that RNA for those muscle-blind protein, for the targets that muscle-blind can target, do we rescue splicing? And we can see that we can rescue splicing. So what we measure are 6 different muscle-blind target mRNAs, and we look at the splicing effects on those. And on the y-axis here, we have an aggregate or composite score. And we can see in a dose-responsive manner, the more compound you apply, the better the rescue on splicing. And we looked at this in 3 different patient cell lines, and we saw the same effect. And we looked at this with multiple of our compounds, and we see the same effect. Moving on to a more complex system, we use what they call an HSALR mouse model. This is from the Thornton lab. And we use this mouse model that contains these CUG repeats within skeletal muscle and asked whether our compounds can rescue the splicing defects within those skeletal muscles. And we did this same sort of assessment as we did on the cell. So we dosed these animals 5 times daily with our compounds over 5 once a day for 5 days, sorry. And we then took calf muscle cells, we isolated RNA and asked if splicing can be rescued. And it can be rescued. These are 2 different doses of the same compound, and you get more rescue of these splicing targets the more compound you apply. And so that was pretty exciting for us. And so lastly, we asked in that same mouse model, can we rescue the myotonia that these mice exhibit? So these mice have a tough time relaxing their muscles. And so what we did was we anesthetized the mice after that same dosing regimen. And we asked whether after a stimulation, how long does it take to relax the muscle? And we saw that our compounds brought that time to relax down from 1 second to half a second for the lower dose and down to just a fraction of a second for the highest dose. And so those are all the data that we'd like to share with you today. We are really excited about this program. We're right now focusing on combining oral bioavailability with a very potent molecule. We're making really steady progress on that. And we are hoping to file an IND first half of next year, which means we'll be filing paperwork to the FDA with the hopes of getting in the clinic about 9 to 12 months after. And we would like to thank everyone who's contributed to this project. We'd like to thank everybody at this conference who's given us advice and reagents and mice and cells, and especially everybody here for being willing to talk to us and letting us know the patient viewpoint. Thank you.
Thank you very much. So this concludes the session. Maybe we can say thank you and a round of applause to the presenters. And then, of course, we will commit to stay in touch with these wonderful folks, right, and arrange webinars, longer webinars, where you will actually have an opportunity to ask questions and interact in a Zoom format live with those folks. So thank you very much. Thank you very much for sitting through these technical presentations, and catch us if you didn't get anything. Catch us in the hallway, and we can try to explain further. Thank you.